Welcome back, friends. In this video tutorial we'll be talking about what are called transcription factors. Okay, so transcription factors. Okay, now what are transcription factors? Now transcription factors are protein molecules. Mostly they are made up with protein molecules, and they are found to have very important effects in eukaryotic transcription. So related to they are related to eukaryotic transcription. Okay, transcription. Okay, now before understanding the eukaryotic transcription, you must have a clear cut understanding of some important concepts of cell and of eukaryotic DNA and their transcription. So let's begin with that. So let me take color here. Let's say this is let's say here it is a structure of a eukaryotic gene, and you need to know the structure here. So let's say this is a eukaryotic gene, eukaryotic DNA, for example. Okay, so this is a eukaryotic DNA. Now in this DNA, there are having 5 prime sequences, and obviously there are 3 prime sites. Now there is a region in between this place which is called a promoter sequence, right? Let's say this one is the sequence. It is called promoter sequence. Now it is a very important sequence because from this particular region DNA polymerase RNA polymerase can come and sit onto this region. It will initiate the transcription, right? Now transcription means simply the production of RNA from the DNA. So if this is the DNA and here it is the promoter, RNA polymerase can come and sit here and they will transcribing the DNA into RNA molecule. Now except for this promoter, there are 2 more sequences that are found in eukaryotic DNA, which is not found usually in prokaryotes. Now let's say here further upstream of that we are having what is called upstream element. So let's say this is also called upstream element or upstream control element, or UCE. Okay, so this part is called the upstream control element, which is present further upstream region of this promoter. Now something which is going toward this direction of promoter is called upstream, and something that is going towards the 3' end, it is called the downstream in this case. Okay, and obviously there are something more that are present in the downstream region of the promoter. Let me draw them. Now here it is. Those are called also, sometimes they are present, they are called the activator sequences. Sometimes they are present, sometimes they may not, but they are sometimes called activator sequences. Okay, so sometimes they can present, sometimes they cannot, but that's the traditional structure of a system, okay, of a DNA of eukaryotic organism. Okay, now what is important about that, for the transcription machinery, what we need to do, we need to have a RNA polymerase. Now RNA polymerase to sit onto the promoter, and then they will open this DNA, and then the transcription will begin. For these purposes, that for the purpose of this transcription and the opening of the DNA, we need what are called the transcription factors. There comes the importance of transcription factors, right? Now usually what happens, RNA polymerase in case of prokaryotic cells will sit and they can initiate the process, but for eukaryotic system they need the importance and presence of accessory proteins, which are transcription factor. Now usually there are protein molecules present and associated with various types of transcription in eukaryotic system. Depending upon the type of polymerase they are acting with, the name of transcription factors always varies. Now in eukaryotic cell we can find 3 different types of polymerase to work on. So the polymerase that we get for eukaryotic cell, let me write it here. So the polymerase, let's say polymerase that we get, there are different types of polymerase. So one type of such polymerase, let's talk about it. The first type is called the polymerase 2, which is the basic type of polymerase, which is the major type of polymerase that we can find, because this polymerase 2 is responsible for the transcription of mRNA molecule. Remember, we are talking about transcription means we are talking about production of production of RNA. And when we talk about RNA, there are 3 types of RNA. One is mRNA, which is the messenger RNA, which is having all the codes for the protein translation. Another one is the rRNA or ribosomal RNA, which is the structural components of ribosome. And the third one is called the tRNA or transfer RNA, which is required to carry the amino acid sequences during the translation event, right? So there are three different polymerase for transcribing each of these RNAs. The polymerase 2 usually used for mRNA synthesis, and we know that mRNA are required most of the time. That's why this is very important. And there is another which is called the polymerase 2. Now this polymerase 1, polymerase 1 is responsible for the transcription of this for rRNA components. And there are also third kind of polymerase which is called the polymerase 3. Now this polymerase 3 is important for transcribing tRNA sequences. Okay, so these are the three different polymerase. But whatever polymerase that we get, it will require certain type of eukaryotic transcription factors, right? Now for polymerase 2, there are some certain transcription factors that we can get, and those factors are namely transcription factor 2. And transcription factors attached to polymerase 1 are called transcription factor 1, and transcription factor attached to polymerase 3 activity are called transcription factor 3. So the naming of transcription factor again is depend upon or depend upon the type of polymerase they are working with, right? So for polymerase 1 it is 1, for polymerase 2 it is 2, for polymerase 3 it is transcription factor 3. Now these transcription factors are again having different types of proteins. That means for transcription factor 2 they are having many. So there are transcription factor 2A, 2B, so like that, 2A, 2B, 2F, 2H, and there are a lot more transcription factor 2s are available. Similarly, for transcription factor 3 also there are transcription factor 3A, 3B, 3C, and also on. For transcription factor 1 also they are having different varieties, right? So these are the variations of different proteins that are available. Now another type of transcription factor, let me talk about the differentiation of the transcription factor. Now this type of differentiation that we get is based on the type of polymerase they are working with, but also depend upon the action and the application of transcription factor. We can also divide transcription factor into two different sections. So let me write it here. So let me write transcription factors. We can divide into two different sections. One is generalized transcription factors, sorry, generalized transcription factor, or say, let me take the dark color. Okay, it is called the generalized transcription factor, or GTF. Another one is called the specialized transcription factor, or STF. So there are two types of transcription factor depend upon their use during the transcription, right? Now these generalized transcription factors are those type of transcription factors that are normally found inside the cell all the time. But specialized transcription factors are those transcription factors that are only found when the cell require a specific type of gene expression. For example, in normal conditions, which are called the household or housekeeping genes, which are the general normal genes inside the cell which is need to be transcribed all the time, there are transcription factors associated with these genes, and those transcription factors are termed as GTFs. But there are certain genes that are present inside the cell which the cell only require to express in a certain environmental conditions in a certain amount of time. Now those transcription, for the transcription of those genes, we require specialized transcription factors, right? That's the difference between generalized and specialized transcription factor, right? So that's it. Now this TF2, TF1, and TF3, all of them that we have just discussed, all of them are the part of normal GTF. So all of these things are the part of GTF systems, and for the STF, or specialized transcription factors, there are other type of factors. Okay, now let us talk about how these transcription factors make this transcription possible, and what are their importance during this process. So now let's say, here let me draw the gene again. So let's say if this is the gene, so if this is the protein, let me draw. So if this is the DNA sequence, now there are a promoter region. Let's say here it is the promoter region. Let's change the color with this green for the promoter sequence. Let's say this is the promoter sequence, and this is the sequence where the RNA polymerase will sit. But RNA polymerase won't get a perfect signal to sit at this particular place until and unless these transcription factors inform polymerase to do so. For example, here we also know that further upstream of it we are having upstream control element. So let's say this is the upstream control element, or UCE, remember, and this was the promoter region. So let me write, this is the promoter, right? So this is promoter, and this is upstream control element, or UC. Now this transcription factors, the first process of during the eukaryotic transcription is the binding of transcription factor. Let's say here say this one is the transcription factor, so TF. So this transcription factor start to bind with this upstream control element. Now another transcription factor come and it binds with the polymerase promoter region of that. So this is suppose another transcription factor. Let's say it's a transcription factor X, it is a transcription factor Y. For example, transcription factor X attached to upstream control element, transcription factor Y attached to the promoter sequence. Now they will have a crosstalk between themselves. Now as they are having the crosstalk between themselves, they will recruit RNA polymerase to the scene. Now let's draw the RNA polymerase. Let's say now RNA polymerase, so here says RNA polymerase is there. Now as there is a crosstalk between TFX and TFY, they'll recruit this RNA polymerase to the place. So now say RNA polymerase will come, it's a large component enzyme, it will come and sit onto the promoter, then it will start transcribing this DNA into RNA component. Okay, so that's the process of how it's done. Now remember, in this case the transcription factor X and transcription factor Y, whatever we are talking about, all of them are of generalized transcription factors. But now sometimes suppose there is a gene, suppose there is a gene present, so let me drag it further. Suppose again, let me draw this for you. Let's say yes, this is a DNA and there is a segment, again promoter region is present somewhere there, so say this is the promoter segment, and upstream control element is also there, so say this is the upstream element, it is present. But so let me write UCE, and this is the promoter sequence. Okay, now let's say here it is the gene, the rest of the part downstream of the promoter, say this one is a part of the gene, right? So we need to transcribe this gene. Now all the time inside the cell this gene is not transcribed. The transcription of this gene is usually blocked inside the cell. Now suppose there is a temperature change in the environment. So if we consider this is inside a cell, so let me draw the cell boundary first. So if we draw the cell boundary here in this case like that, and obviously like that, say this is the cell boundary, and inside the cell we are having the DNA. Now there is a sudden change in the temperature outside. For example, temperature increased from 27 degrees Celsius to 39 degrees Celsius. So there is a sudden increase in temperature outside. As a result of that, inside the cell, if the cell need to maintain its homeostasis inside, what it need to do, it needs to synthesize some of the important proteins which are called heat shock proteins. Remember, heat shock protein, or HSP, which will help this cell to combat this high temperature situation, right? So if the temperature increase like that, in those conditions there is a gene for this HSP. Now this gene is encoding a protein of HSP. Now they need to transcribe this gene, but when the temperature was normal, like 25 to 27 degrees Celsius temperature, cells don't need this HSP, so the transcription of this HSP gene was previously blocked. But now as the temperature is up to 39 degrees Celsius temperature, it need to reinitiate the transcription and translation of this HSP gene. So for that purpose it need to switch this HSP transcription on. Now for the switching of this HSP transcription, they require some certain transcription factors, and these transcription factor in this case are called specialized transcription factor, because remember this is a special case where the temperature change occurs, so we require specialized transcription factors. So there specialized transcription factor will come and it will sit and attach, bind like this. Remember, specialized transcription factor is having a structure like that. Okay, so let me remove this part first. Okay, so specialized transcription factor will be there. So let me draw this. Here it is a specialized transcription factor inside the cell, and also there are some generalized transcription factors. Now I've told you that specialized transcription factor always work at a special conditions, but there must be generalized transcription factors always present inside the cell. So here always generalized transcription factors are present. Now these generalized transcription factors will come and attach to the promoter region first. So GTF, so let me write GTF first bind to this promoter region, and obviously they can bind to further upstream or downstream of the promoter also. So let's say here is another GTF, bind with it, then specialized transcription factor will come to the play. So specialized transcription factor will make a structure something like that, because specialized transcription factors are having two important segments. So let me draw these parts. Let's say here let's say this one. Now this part of the specialized transcription factor, this is called the DNA binding domain of specialized transcription factors. So this is DNA binding domain of specialized factor, and this part is different. It is called, so this segment is polymerase binding domain, okay, and protein binding domain. Okay. Now, as they are binding with the generalized transcription factor, they will recruit DNA polymerase to the scene, right? Sorry, here RNA polymerase. So they will bring RNA polymerase to the scene. So RNA polymerase will come and they will be sitting onto this place now. Let's say here this one, and here it is, the RNA pol. They will sit onto the place and they'll start the transcription of this HSP. Now as they'll start the transcription of HSP, so let me take it further, yeah, let's like this, okay. Now as they will start the transcription of HSP, further after some time what we get, we'll be getting more and more of this HSP, sorry, not this one, yep, this one was right. Anyways, so more and more HSP molecules in our hands. So here are more and more HSPs, right? So these are the HSP, mRNA will be made, and from this mRNA HSP proteins will be made, right? So this is how they control the whole process. So we can see the how these transcription factors are controlling the expression of certain genes inside the cell, and obviously during this expression process they require many more factors. They are also called, some of them are also called mediators, right? So let me write it here. So among this transcription factor, so let me write the types of TF. So depending upon their functionality, we can have different types of transcription factor. One type which are helping them to activate a gene, right? Or we can say to activate transcription. So let me write it here. So activate a gene or transcription. There are certain types which are usually to what we can say prevent transcription, right? So there are two types of uh, transcription factors that are found. Now, usually those one which actually works to activate some genes are called activators, as we have seen in this case. In this previous example we've seen some activators which activates the expression of HSP gene. There are certain types of transcription factors which can downregulate the expression of genes. These are called deactivators or inhibitors, right? Now, in both the cases, transcription factors can work alone, or sometimes transcription factor needs some other accessory proteins to function with them, right? So in those cases when transcription factors need some accessory proteins, the accessory proteins that can function in this case are called mediators. So let me write it here. So from here, so there are also mediators that are present. So let me write the example of mediators. So let's say here it is, there are mediators. Now, mediators will work with transcription factors. So let me write it here. Say here it is TFs to bring either of the two effects. So they will interact with them to bring either of the two effects: activation of a gene or inhibition of a gene. So this is an important concept, right? So mediators are those molecules. And if I draw mediators, how mediator will look like, let me draw this for you. Okay, so if I draw again a gene, so how mediator will look like. Now let's say this is a gene. Now after this is a DNA, for example. Now after the binding of everything, it will look something like that. Let's say here it is. So let me draw the promoter. Let's say this region is the promoter. So let me write it here. It's a P, and polymerase is sitting onto it, and other factors. So let me draw the factors. So these are the transcription factors. Remember, TFs. So let me draw there. All of them are TFs there, and obviously uh, polymerase is sitting here, right? This is Pol, say Pol 2 in this case, and there are some certain activators. So let me draw the activators. So say these are the activators, right? So these are the activators that are attached, and mediators are present at the middle. So let me draw the mediator. If I draw the mediators in this case, here comes the role of mediators. So you can see mediators is actually holding activator and other TFs together. So this middle part is role of mediator. As the term suggests, they mediate the process, right? So here comes the importance of mediators. And also there are mediators like different histone modification agents. So mediators called different types of histone modifying enzymes, right? Histone modifying enzymes like histone acetylase or histone acetyl transferase or HATS, and histone deacetylase or HDAX, and those are different examples. Histone methylase, so like DCM, DAM, and so on, right? So these are the examples of different mediators that are also found inside the cell. So transcription factors are protein molecules which are helping to transcribe certain genes in certain conditions or sometimes in normal conditions. They will require the functionality of some mediators for achieving the function, either activation of the transcription or inhibition of the transcription. Okay, so that's it, and I hope that's helpful. Thank you.